This is a demonstration of the TCGA Analyzer software. TCGA Analyzer is software that demonstrates data mining concepts and offers a tool for human genome analysis. TCGA Analyzer uses the Eclipse-rich client platform paradigm. Essentially, it is a set of custom plugins for the Eclipse platform that run as a custom application. This gives the developers significant user interface capabilities for free and allows the developers to concentrate on the problems at hand. So imagine Eclipse reskinned as a new application. TCGA Analyzer contains several panels. On the left side are the Data Explorer and Genome File Parser panels. While the Data Explorer panel has yet to be fully realized, the Genome File Parser allows the user to load genome data into the database. Next, we have the Heat Maps panel. The Heat Maps panel offers two heat maps, the main heat map tab, which offers the user an interactive way to drill down into genome data, and the density heat map tab, which offers a hierarchical approach to viewing the genome data. On the bottom, we have a tabbed view, which provides an integrated web browser, the raw data browser, and an experimental 3D heat map, which unfortunately was not ready in time for this demo. In order to view the genome data, the raw data which is downloaded from the Cancer Genome Atlas as text files must be parsed and loaded into a Postgres database. The parsing utility will crawl a directory structure looking for the TCGA formatted directories and files. All the user needs to do to import one or more files is to locate the base file or directory in the file tree, right-click on the file or directory of interest, and select Parse Genome Data File from the pop-up menu. The file or files are then parsed and imported into the Postgres database. The user is then notified when the parser has completed. A well-organized heat map can help researchers easily identify points of interest. The most challenging question then becomes, how do we arrange genes in the heat map? In the beginning, we can use some automatic technologies to group genes which may have similar behaviors or functions, or use well-known gene knowledge to decide the order of genes in the heat map. Once we generate this kind of heat map based on this knowledge, we can use knowledge from the researchers to rearrange genes in the heat map. We can record all kinds of arrangements in TCGA Analyzer and can apply this information to further analyze other data samples from the TCGA data portal. Using this, a researcher would typically use the heat map to navigate the genome data. Points or areas of interest could be located on the heat map by looking at a gene that had, say, different values for different sample values. Or perhaps a point of interest might be a gene that expressed high values throughout all the sample values. By hovering the mouse over any spot on the heat map, the researcher gets both the gene ID and the sample ID shown in the window on the right side of the screen. The selected gene can be added to a list of favorites for further study. The integrated web browser contains the Annotation and Evaluation tool, which is accessed by double-clicking on a gene in the heat map. The Annotation and Evaluation tool consists of three parts: the gene report and summary, the annotation chart, and the related gene list. The gene report and summary shows the user input gene from the heat map. Due to the large number of databases and associated IDs, the conversion of gene identifiers is one of the initial and central steps in workflows related to the genomic data analysis. In this project, we use the TCGA genomic data, or Affymetrix ID. In order to retrieve related messages, we convert the Affymetrix ID to an Entree ID and David ID. The left-hand side shows the Affymetrix ID format, and the right-hand side is a David ID format. The second section of Annotation and Evaluation tool is the annotation chart. The annotation chart is an annotation term-focused view which lists annotation terms and their associated genes under study. If input genes are associated, we should receive many annotations. The annotation chart consists of category, term, count, percentage, p-value, and gene combinations. The third part of the Annotation and Evaluation tool is a related gene list. The related gene list provides two pieces of information. The first one is the most frequent gene in the combination of the annotation chart. The second is a related gene list. This list uses the most frequent gene above as a center point to find related genes. The researcher can double-click on a point of interest on the heat map and get more information in the Evaluation and Annotation window below. In the Gene Report section, we can click the yellow link, or David ID, and the application will link to David Gene Report. The David Report consists of a gene summary, official gene symbol, and disease category. Back in the Annotation section, we can click the green link and the application will link to the EBI ancestor chart. The ancestor chart provides a concise view of the relationships connecting the gene ontology terms. Finally, back in the Related Genes section, we can click on the yellow links to access more David data for the genes related to the most frequent gene. We can use multiple scales to measure the values of gene expressions and build scale linking in TCGA Analyzer. For example, at the first level, we see the heat map of all genes whose measured value falls into the 0 to 100 range. At the second level, we see the heat map of all genes whose measured value falls into the 25 to 50 range. And at the third level, we see the heat map of all the genes whose measured value falls into the 25 to 37.5 range. In this way, researchers can observe patterns in different levels. In addition to the interactive heat map, TCGA Analyzer also offers a hierarchical or multi-scale heat map for use in sorting out large pieces of data. To do this, the researcher can select an interesting area using the Selection button. Once the area has been selected, the selection can be applied, and the heat map zooms into that area. This can be done multiple times to zoom in as far as the researcher likes. The forward and back buttons can be used to navigate between different zoom levels. In addition to a single area select, the researcher also has the option to select multiple areas of interest.